Hello guys, welcome back to my channel. Today I will explain about something that's very important, a type of bacteria known as Salmonella, and I'm writing these things in my note because I see it as a better thing. It's simple, easy, and you can add pictures, so I'm writing the informations in my note. And the note, yeah. What's Salmonella? First of all, Salmonella is a type of bacteria. We have about more than 1,500 types of Salmonella. And it's a medical important bacteria that causes septicemia or sepsis, and it causes gastrointestinal infection. And it causes severe diarrhea, and it's transmitted through raw meat or infected meat by eating infected meat, and of course milk, animal products. Yes, it's found in, or it's transmitted through raw milks and meat, eating meat, raw meat and uncooked meat. For sampling for Salmonella, we take blood cultures in special bottles, of course, and we incubate them. After that, we culture just as other types of culture. We culture the blood, and the most common type is the gastrointestinal infection, and it causes diarrhea. We take a stool culture, and we use some types of medias. XLD is the most, SS Salmonella Shigella, DCA, but DCA is not that common in veterinarian uses, but I've wrote it. In veterinarian uses, we use just XLD. Of course, we use McConkey too. McConkey is important for gram-negative bacteria. First, the bacteria is facultative anaerobe bacteria, and the selective media is XLD, xylose lysine deoxycholate agar, and SS agar or Salmonella shigella, and it's very famous. There's growth on McConkey, and it's non-lactose fermenter, it's non-lactose fermenting colony, and growth on DCA agar, and it's pale yellow and non-lactose fermenter. The colonies, there's an important point, are sometimes black center. What mean black center? In the center we have a black dot due to the production of H2S, hydrogen sulfide gas. Hydrogen sulfide gas due to the production of hydrogen sulfide gas. And we can do triple sugar iron test, TSI, and in my opinion, triple sugar iron test is very important. In our lab, one of the most important tests are done, of course, catalyze, oxidase, gram stain, gelatin hydrolase or TSI. TSI is one of the most important tests to detect enterobacteriaceae membranes and some gram-negative bacteria. As we said, it's H2S production. It produces H2S Salmonella. Generally, we have typhi, Salmonella typhi and paratyphi, and other species. Yeah, it's H2S positive, and in TSI triple sugar iron, alkaline acid, alkaline slant and acid bath. So what we will write in our report, we will write K over A, and positive of gas production, and H2S. In H2S, black is seen, a black substance, a black color, the media will change to black. This is considered H2S positive. And alkaline slant, as we said, so K over A, and we will write the gas production. As we know, in some bacteria, we know, for example, E. coli, it's easy to find on TSI. It's just A over A or acid over acid acid slant and acid butt, and it's positive gas production and no H2S, and it ferments glucose. E. coli, for example, is one of the easiest bacteria to detect on triple sugar iron. And we have here, it's triple sugar alkaline and acid bus. We have one important point: production of gas is positive except Salmonella typhi. Salmonella typhi doesn't produce gas. Okay, and H2S positive for both Salmonella typhi and other species. Citrate positive. We have a test called citrate test. We have a special media. We stab, just like the TSI, we stab into the butt and streak the slant. We incubate. The color will change, just like blue. Will change from, it looks like green, and it will change to blue in citrate test. So Salmonella typhi is negative for citrate test. So this is the difference between them. Salmonella typhi is negative for citrate test, and paratyphi, of course, are citrate negative. And glucose fermentation with acid and gas production. As we said, in gas production, Salmonella typhi does not produce gas. We said lactose fermenting negative, lactose fermenting negative, urease negative. That's very important. That's one of the difference between typhi and other species. Indole negative, mannitol fermentation positive. Here I have found some pictures for you. The pictures are the gram stain results. In the first picture, as we see, it's a bacilli, or we can say rot, gram negative rot. This is the gram negative rot. Some pictures of gram stain, 2 pictures. We may have different results, but they are the same gram negative. Look, for example, here it looks different, but no problem. The right thing is it's gram negative bacilli. Sorry, I'm a little bit sick today. The weather have been changed in our country. Here we have Salmonella on SS agar or Salmonella shigella agar. We see the black colonies. For doing gram stains, we take individual colonies, of course, or biochemical tests, we take individual colonies. As you see here, a black colony indicates due to the production of H2S or hydrogen sulfide. And this is Shigella. The Shigella color is easy to find. It's different, but in other videos I will explain about Shigella. And I will have a note at the end. As we see, this is Salmonella typhi. Salmonella typhi and other species of Salmonella are all non-lactose fermenter. As you see, you can do motility test, but it's not that common, motility test for Salmonella. As I say, TSI, Simon citrate test, mannitol fermentation, those are the most common tests in my lab. I can't say in every labs, but in most labs of vet, we do those tests. And it depends on your country, which type of bacteria are common, which type of infections are common. As you see, this is non-lactose fermenter. What we mean by non-lactose fermenter? It doesn't ferment lactose, and it will not change the color of the media. It will take the color of the media, and that's how it looks like on McConkey agar. As I have to give a point, urease test, we have indole test. Indole is one of the MVIC group test. MVIC, we have a group of tests called the MVIC test, and I will make a video about it. MVIC test and urease test. We how we prepare urease tube or media. Of course, we have urease base media, base agar, and we add the wanted amount of water and media. We mix it and we just do just like preparing other medias. We have something called the urea. What's urea? It's just like a reagent. It's the same as blood agar. How first of all we just prepare the blood agar base or nutrient agar. After that, and we will wait to till the temperature reaches about 40 Celsius, and after that we will add the blood. So this is just like blood agar. We will prepare the base of the urease agar, and we will add a reagent called urea. But how will we add it? There is something called filter, just a filter, or a syringe filter. We take the five, it depends on the amount. We take the urea reagent and we attach a filter to the end of the syringe, and we put it in the media or in the prepared agar solution. Of course, we mix a little bit, hand mixing, hand twisting the flask. We mix, and this is called the urease agar. Of course, you inoculate the colony into the tube. After that, you incubate and wait for the results. This is about ureases. And indole, as I said in the MVIC tests, I will explain about it. It's different type of test for enterobacterial membranes. So thank you guys. Hope you get benefit.